So
Yeah,
first,
Barbara.
thanks Professor Wang and Dr. Xingang Li for organizing this meeting as well as inviting me to give a talk. I think that my talk is different from the previous several professor labs. They did a very deep study on specific cases. So in my talk, I'm going to talk how could we study the mechanism where the RNA glycosylation could be. So it's kind of like a hypothetical theory, not a exact, without much of the experimental to prove. So I'm going to talk this why glycosylation matters. Then I'm going to talk RNA glycosylation is extension of the central dogma. The Paris central dogma, which is coined by Professor Wang. And then I'm going to follow this to the rest of the talk. So why glycosylation matters? The biggest reason is that glycosylation is play different diverse role in biological functions of the cell. For example, it can mediate cell-cell function and communication in different diseases, as shown in previous several talks. Here I want to show you why we call the paracentral dogma, because in the glycosylation is not driven by genetic code. Instead, it being by the non-template driven process. For example, glycosylation is formed by the different enzymes like glycosyltransferase or glycosidase. I'm not going to talk this in very detail. However, as we know, RNA glycosylation also played a significant role in cell-cell function analysis. However, because this is the early stage of this field, we do not know much about the functionality as well as how the RNA glycosylation is formed. So in this talk, I'm going to show you my hypothetical pathway where the glycosylation could happen in the cellular machinery. So this everybody, I guess, is very familiar with, like for the N-glycosylation, left side, O-glycosylation. Why is this? I wanted to show this because in the RNA field, currently we thought RNA glycosylation may follow similar paths of the protein glycosylations. So I'm going to have some evidence from the other labs publication to show this. Why we call the paracentral dogma, because the protein glycosylation occurs via non-temperature driven mechanism, as I said in previous slides. It made by coordinated enzymes action by like glycotransferase, glycosidase, and it happens for the protein in the ER, then in the Golgi for the N-glycosylation. Of course, the O-glycosylation is happening in Golgi apartment as well as mitochondria. So I want to use these slides to show you what is the difference between the central dogma concept as well as the paracentral dogma. You can see in the central dogma, we from the DNA transcript and to the MR messenger RNA to in the rough ER, it forms a synthesized protein by translation. It forms a peptide bond. In theoretically, the one gene could form one protein. However, in the paracentral dogma, we can see that in the microenvironment, we have hundreds of the enzymes, like we said, glycotransferase, glycosidase. It forms the N-link glycosylation as well as the O-glycosylation in ER or Golgi. In theoretically, one gene could have different glycoproteins because we could have a different glycoform in disease or other stages. So as of today, RNA glycosylation, what we know is the tRNA uridine. This linkage has been identified by Dr. Shi and Dr. Flying's lab. They showing that in the tRNA uridine, under the catalyst of this gene, it forms the ACP3U, and in this this linkage, it could, under catalyst of the OSC subunit, people suspected it could be STTA3A or OST STT3B. It could form the ending glycan linked to the ACP3U. So this is the only non-linkage of the RNA glycosylations. Of course, we identify other RNA glycosylation, but it's not clear what the linkage is, as well as what is the site it could be. Once the glycoRNA formed intracellular, it could transfer to the cell membrane from the EV and communication with the other cells, such as the cyclic. And this is some of the work you already heard from Dr. Len's his PhD work, Nature Communication work, as well as later ongoing work. So for this, I wanted to, of course, we already have some evidence, RNA glycosylation from different labs. So here I want to show you potential hypothetical synthesis of like tRNA, how it could form on the cell membrane. It could, starting from the nucleus, from DNA transcript to RNA in the cytoplasma or some cellular machinery, that form the glycoRNA, then transport the surface and communicate with other cells. So here is you can see, you already saw these slides in Dr. Len's slides, and this is from Dr. Fein's lab, showing that using the metabolic labeling, it can show that glycan salic acid can be incorporated to the glycoRNA. So I'm going to skip this. One recent study from Jiangnan Daxue, Dr. Liu's lab, showing that if we regulate the protein glycosylation, then we will find that the glycoRNA glycosylation will also be effect. Its conclusion is that the surface glycoRNA might be linked to the protein glycosylation pathway. So the next question I want to ask, so which RNA could be glycosylated? So here is like several non-coding RNA which is abundant in the cells. So I'm going to skip these slides. However, I want to show you if we localize where those small RNA inside the cell, it shows that in nucleus, it could have a snRNA, tRNA, RNA, as well as link RNA. In the cytoplasma, like the rough ER, we can see the microRNA as well as the microRNA inside the cytoplasma. Some of the RNA might be located in the mitochondria. So why this is important? Because our hypothesis is if the small RNA located to nearby or inside the glycosylation machinery, it could be glycosylated. So these slides basically show you what a post-transcriptional modification has been found in those RNAs. You can see there is like pseudo-uridine methylation, as well as other different types of chemical modifications. So one thing I wanted to emphasize here, for the glycosylation, we found those like link RNA, microRNA, SNO, and as well as snRNA, tRNA has been N-glycosylated as well as O-glycosylated. However, for the messenger RNA, there is no evidence so far whether it can be glycosylated. And rRNA, we also need more evidence to show whether this exists. So next few slides are going to show you a possible glycosylation pathway in those different RNA species. And this is a tRNA which has been determined by the many labs, showing that ACP3U is one side for the tRNA glycosylation. However, we guess there is many other sites it could be glycosylated. So what are those sites remain to be discovered. So these slides basically show you the potential mRNA glycosylations. So I just using the biogenesis of the mRNA to show you where potential place it could be glycosylated. of course, we do not know yet. However, my guess is it could happen on the nucleotide, or may happen on the poly-A tail. If the mRNA can be glycosylated, what are potential functions? Because the mRNA translate to the protein synthesis. Therefore, if indeed there is a glycosylation on mRNA, it could affect the mRNA stability, or protein synthesis efficiency, or gene expression, etc. So next, I wanted to show you a little bit of my hypothetical pathway for the microRNA synthesis. The microRNA starting from a nucleus, from the primary mRNA, then through the dorsal DGGR8 to form the primary microRNA. Then inside the nucleus, we think it could be all glycosylated. This remains to be discovered. Once secreted to the cytoplasma, it goes through the could be on the ER. So it could be unglycosylated, as well as can be galacsylated mucin type oglycolations. Also, we found that the mRNA could be on the mitochondria, as we show in the previous slides of the cellular location. It could be also oglycolated. So, however, this type of work hasn't been proven yet. So far, we only know RNA can be N-link glycosylated and O-link glycosylated, not a glucan or other type. So basically, I'm going to skip this. Is similar to summarize previous slides for the synthesis. So there is potential functional rule change if we have a microRNA glycosylation, and in the disease, how those glycosylation change could impact the gene expression, stability, silencing efficiency, etc. So those is our open question need us to tackle. So I guess in China there is many labs starting to work on this. In US, many labs also did an excellent job on these directions. So next, how about the RNA? Can it be oglycolated? This is still ongoing, so I'm going to skip this. So next one is a yRNA. So I wanted to mention all of those schematic structures from my digging the literature to find out how it could be, what the process is. So it's not validated yet. Next, I wanted to show you what technology can be used to studying glycoRNA. So as Dr. Then summarized, this method, DRFET. So this can be used to image the RNA, and the ARPLA also can study the cell-related cell membrane glycoRNA. In the first approach, this is from our labs, SPCGRNA or TINO-RNA. Basically, can use this to enrich the glycosylated RNA species, meaning that you can detect whether this RNA has been formerly glycosylated. RPAL can be used to detect inside, as well as what the linkage between the sugar and the RNA substrate. The other key method, like we mentioned before, the metabolic labeling, as well as a CO2 lectin-based detection. So this, you can find the papers which describe how to using those approach to detect the glycoRNA. So I'm going to very quickly go through this. This is a method called SPCGR, basically using the GAO stands for galactooxidase to oxidize the gal or GalNAc for conjugation on the hydrogen beads, then using the PGSF to release the N-glycan. So this is basically the data showing this method is valid. Is the RNA we identified is from the RNA, not from protein or other contaminations. So this is for now. This method, the advantage is you can study cell culture, you can study the tissue from patient sample. You can also using the human body to study the N-link glycoRNA. Similar, this method, tRNA, basically look at the TN containing O-glycoRNA. Because we only have this GalNAc or O-glycosidase to remove the T antigen or T antigen from the substrate. Therefore, currently for O-glycosylation, we have limited type of O-glycoRNA can be studied using this approach. So we use tRNA to study the pancreatic cancer cell line. It shows that there is different types of RNA present, and by looking the normal cell versus PDAC cell, we find that some certain microRNA has been significantly increased in the cancer cells. So also we touch base a little bit about what could be the O-glycosyl to transfer the oligosaccharide precursor to the RNA backbone. And using the NGI, we can understand how this could whether the glycosylation transferase is similar to the protein OST transferase. It shows that maybe they share the same OST subunit, this STT3AOB for the RNA glycosylations, but this may not be true. I guess the STT3A may partially participate in the RNA oligosaccharide precursor transfer. The other subunit maybe need to be determined. So finally, I wanted to show you a functional investigation in terms of glycoRNA. So this is basically a workflow show you how can we do the functional study. As I show you in the previous slides, if we only know the RNA glycosylated, however, we do not know where the glycoside is. So basically, we have these two software called glycan SRA or PON glyRNA to predict the site N or O-link glycoside. Then once we know where the site is, we can do the mutation, do the bunch of the study, like cell mouse model to study the function of those the glycoRNA. I'm going to give you a little bit of the example in the next slides. For example, in the PDAC, we found that micron 21 has a glycosylation change in the PDAC. So we using the software to predict the site like this has been 2 glycoside. Then we can do the mutagenesis on this T to A or A to G as shown here. The glycoside will be removed. So once without glycosylation, this is only pure RNA precursor. So we do this apoptogenesis study of the PDAC cell, showing the green, showing that once we abolish the N-glycosylation, it will affect the apoptosis rate. Similarly, it shows that the cell proliferation has also been slightly affected by remove the N-glycosylation site. Finally, I wanted to show you what our lab is ongoing work to tackle where the glycosylation happens. For example, if we wanted to study the microRNA glycosylation, where this glycosylation can happen, we can using the metabolic labeling, then doing the two label on the cell culture, then detecting RNA and using the confocal or other approaches. So in terms of future direction and what is the glycoRNA related to the glycomedicine application, we think that first of all, we need to know the site. As of today, we only know the ACP3U is a definite site. How about others? So once we know this, we can do the mapping of the RNA glycosylation. What is the other thing is what is the enzymes specific to the RNA glycosylation, including glycotransferase or add the carbohydrate residue to the glycan, etc. So biological studies as well as glycomedicine applications using the RNA technique as such as diagnosis, therapeutic, etc. So to my end of my talk, I hope through my slides you know that glycosylation is a paracentral dogma, is extension to the central dogma. Secondly, RNA glycosylation has been demonstrated. However, there is many of the unanswered questions: how the synthesis is, where is the machinery of the glycosylation. Finally, if we understand glycosylation, it is a key for glycomedicine for, like I said, biomarker diagnosis or therapeutics. So finally, I wanted to acknowledge the funding from Shantou University Medical College and as well as the first hospital affiliate hospital of the SUMC. I want to advertise, like Professor Wang said, if you want to join Institute Glycome study, please send your CV to us. We recruited postdoc as well as the PIs. So postdoc have the different location. You can work in the SNO or Yuanqi in the Suzhou, or you can work in Shantou Medical College. With that, I thank everybody online to stay to listen to my talk. I'm going to answer your question after the Professor Gordon Knox talk. Thank you.